At the very onset, I am Dr. Abhijit Majumdar, acting as the Professor and Director of Noida Institute of Engineering and Technology, Pharmacy Institute, Greater Noida. Would like to extend a very warm welcome to all of you to my class today. Basically, we will be studying today about LAL test. Now, as we know, as a student of pharmacy, that we are producing a large number of pharmaceutical preparations, particularly let us talk of the parenterals. It has been seen that there are incidents where there is a presence of pyrogen in the pharmaceutical preparation. That is, the pharmaceutical preparation which you have manufactured, due to improper manufacturing, improper sterilization, improper preservation, there are situations where there is a presence of pyrogen. Now, let us define what pyrogens are. Pyrogens basically are non-volatile in nature. Mainly, they are produced by the gram-negative bacteria. The problem with pyrogen is, whenever we are taking a preparation which is containing pyrogen, a metabolite of the gram-negative microbes, this pyrogen is found to create a fever. And this was utilized to evaluate the pyrogens taking the help of the sham test and the main test using rabbits as an experimental animal. But as because there are lot of restrictions on the use of animals, we basically from the industrial perspective, we have shifted or we have explored another method by which we determine the presence of a pyrogen in the pharmaceutical preparation. And that particular method is known as LAL test. So, LAL test is used to measure the concentration of the endotoxin which is obtained from the gram-negative bacteria. It is nothing but the amoebocyte lysate of the horseshoe crab, which is also called Limulus polyphemus. Basically, it is a perfect solution which we obtain from this horseshoe crab, and moment the main fundamental process is, whenever we are putting a preparation which we want to test for the presence and absence of pyrogen, if there is a presence of pyrogen in the preparation, immediately that will be converted into the sol to gel form. That is, by seeing the consistency after adding the test pharmaceutical preparation, we can conclude that whether the pharmaceutical preparation is containing pyrogens or not. So, addition of solution which is containing pyrogen, which is our test substance which we want to test, to a solution of the lysate of amoebocyte produces turbidity, and if it produces turbidity, it means there is a presence of pyrogen. Now, the rate of this reaction depends on endotoxin that is the amount of endotoxin present in the preparation. It depends on the pH, it depends on the temperature also. So, the rate of conversion from salt to gel, it depends on number 1, the concentration of endotoxin, it depends on the pH, as well as it depends on the temperature. And this test is based on the clotting mechanism of the horseshoe crab. Now, here I will be showing you in a video how we are preparing the LAL test reagent with the help of horseshoe crab. Let us see the video first of all.
The Atlantic horseshoe crab, Limulus polyphemus, has long been valued by the biopharmaceutical industry as a source of critical proteins that detect the presence of harmful bacterial endotoxin. Called Limulus amoebocyte lysate, LAL, these proteins come from the animal's blood cells, amoebocytes, and serve as the foundation of the LAL endotoxin test. In the 50 years since its discovery, LAL has proven to be the most sensitive and effective indicator of endotoxin contamination in treatments like vaccines, IV drugs, and medical devices. At least 10 times more sensitive than traditional test methods, LAL can detect as little as half a picogram per milliliter of endotoxin, equivalent to 1 grain of sand in an Olympic-sized pool. Because it is a natural, renewable source of LAL, the horseshoe crab has become the focus of tremendous efforts to preserve and protect this incredible creature. In 1992, Endosafe founder Dr. James Cooper wrote draft legislation to regulate horseshoe crab fisheries. As a result, South Carolina enacted laws to protect its indigenous crab population. In combination with the efforts of advocacy groups, the laws have served to preserve the animal's natural habitat and prevent their use as bait. South Carolina is now home to 8 island sanctuaries. Conservation efforts have spread to span the eastern seaboard. The Atlantic States Marine Fisheries Commission now mandates that all Atlantic coastal states identify horseshoe crab nesting beaches. Charles River is committed to the conservation of this extraordinary species that contributes to the everyday health and safety of people and animals all over the world. As a responsible manufacturer of LAL, we are proud to play a role in alleviating pressures on the horseshoe crab through active animal welfare campaigns, decades of R&D, and scientific innovation. Each year, our highly controlled and monitored bleeding programs begin with hand collection of crabs, performed by our licensed fishermen in local South Carolina waters. Our technicians practice humane care throughout the health inspection and blood collection processes, and animals are returned to the sea within the same day. Continuing our conservation, we strive to do more with less. We've developed FDA-licensed LAL cartridges that use 20 times less raw material than traditional LAL tests. We are constantly refining our cartridge technology so that responsibility rather than demand can continue to guide our operations. Used in our portable endotoxin testing systems, our LAL cartridges produce point-of-sample results in just 15 minutes. It is critical that we serve as advocates for the humane treatment of horseshoe crabs and strive to achieve balance between our need for this valuable material and the livelihood of the animal that provides it. To learn more about how we are helping safeguard the continued population growth of the horseshoe crab, please visit hsc.seariver.com. Same day. Continuing our conservation, we strive to do more with less. We've developed FDA-licensed LAL cartridges that use 20 times less raw material than traditional LAL tests. We are constantly refining our cartridge technology so that responsibility rather than demand can continue to guide our operations. Used in our portable endotoxin testing systems, our LAL cartridges produce point-of-sample results in just 15 minutes. It is critical that we serve as advocates for the humane treatment of horseshoe crabs and strive to achieve balance between our need for this valuable material and the livelihood of the animal that provides it. To learn more about how we are helping safeguard the continued population growth of the horseshoe crab, please visit hsc.criver.com.
I hope all of you have enjoyed the video. Now, whenever we are talking of this LAL test method, there are basically 5 methods by which we can use this LAL test for detecting the presence of a pyrogen. I am repeating once again. Basically, there are 5 tests which are commonly used to detect the presence of the pyrogen. The first one is the gel clot method. The second one is the turbidimetric kinetic method. The third one, a common variant of the turbidimetric process, a turbidimetric endpoint method. Fourth one, chromogenic kinetic method. And lastly, we are having chromogenic endpoint method. These are the 5 methods which we take the help of whenever we are determining the presence of a pyrogen in a pharmaceutical preparation. Let us see how these methods act upon. In the first method, which we are calling as the gel clot method, the term gives you an idea of the process. The process goes like this. In presence of endotoxin, a proenzyme which is present in the amebocyte lysate, which was earlier inactive, is now converted into an active form, and after being converted into an active form, they are acting on the clotting protein which are present within the limulus amebocyte and converting them into a clot. I am repeating once again. In presence of an endotoxin, the proenzyme which are present in the amebocyte lysate, which was earlier inactive, is now converted into an active form. That is, the endotoxin has converted their inactive form to active form. The proenzyme has been converted to an active form, after which it is acting on the clotting protein present within the limulus amebocyte, and when it acts on the clotting protein, it is converted into a clot. That is, there is a transformation or conversion from the liquid consistency to a semi-solid consistency. That is, there is a change of consistency. So, this is converted into a clot, and moment there is a presence of clot, we will conclude that there is a presence of pyrogen. We will be concluding that there is a presence of endotoxin. That is, the presence of endotoxin tells you, I mean, is calculated or determined by the noting that whether clots are present. When the clots are present, the test is positive, it indicates that there is a presence of endotoxin in the final pharmaceutical preparations. Next, we are moving on to the next method. Now, in turbidimetric method, as I have told you, that there are two sub methods. What are the sub methods? First sub method is turbidimetric endpoint method, and the second method is turbidimetric kinetic method. So, there are two methods. One is turbidimetric endpoint method, other one is turbidimetric kinetic method. In turbidimetric endpoint method, there is a quantitative relationship between the concentration of the endotoxin and the turbidity of the reaction mixture. There is a direct relationship between the concentration of the endotoxin and the turbidity of the reaction mixture produced at the end of the incubation process. Therefore, by noting the turbidity of the reaction mixture at the end of the incubation process, you are in a position to tell how much amount of endotoxins are present in the preparation. The next method is turbidimetric kinetic method. It measures the time needed for the reaction mixture to reach a predetermined, pre-specified value of absorbance. In case of turbidimetric kinetic method, we are measuring the time needed for the reaction mixture to reach a specified or predetermined value of absorbance. We are in a position to determine a initially predetermine the value of absorbance, and we in turbidimetric kinetic method we measure the time required for determining the reaction mixture or the pharmaceutical preparation, to be more specific, to reach that value of absorbance. Now, another way of looking at things is it measures the rate of development of turbidity in presence of the endotoxin. It measures the rate, it is measuring the predetermined rate of turbidity development of the pharmaceutical process. So, by the help of determination of with the help of turbidity measurement or turbidity detection, we are in a position to tell whether the pharmaceutical preparation is containing endotoxins or not. If containing endotoxin, we can also tell the amount. Next is the chromogenic technique. In case of chromogenic technique, again we are having two different processes. One is known as the chromogenic endpoint test, where the term chromo means color. The quantitative relationship between the endotoxin concentration and the quantity of the chromophore released. That is, here there is a link. In earlier one we were linking with the turbidity. Here there is a direct relationship, a quantitative relationship between the endotoxin concentration and the quantity of the chromophore release at the end of the incubation process. At the end of incubation process, we have to estimate the amount of the chromophore released, and we have to link it with the endotoxin concentration. The next method is what we are calling as chromogenic kinetic test. Kinetic means movement. Here in this chromogenic kinetic test, we have to measure the time needed for the reaction mixture to reach the particular predetermined value of absorbance. It is the time needed for the reaction mixture to reach the predetermined value of the absorbance. That is what we are calling to be as chromogenic kinetic test. I hope it is clear to you. So, chromogenic technique, it measures the amount of chromophore released from the peptide by the reaction between the lysate and the endotoxin. Basically, what we do is we are measuring the chromophore. We are measuring the chromophore. In one case, in the chromogenic endpoint test, we are measuring the endotoxin concentration and correlating with the quantity of chromophore released, and in case of chromogenic kinetic test, we are measuring the time needed for the reaction mixture to reach a specific value of the predetermined absorbance. Good. So, this chromogenic technique is based on the development of color after cleavage of synthetic peptide chromogen complex. Finally, the LAL reagent is formulated with synthetic substrate which gives you an yellow color in the case of chromogen when endotoxin activated enzyme acts on that particular preparation. Let us move on to the next slide. Let us see what are the major application of this LAL test, where this LAL test is very commonly applied. Now, LAL test is applied in various pharmaceuticals, whether it is a case of a parenteral or it is a small and large volume parenteral, both we are using. We are using LAL test for biological products like vaccines, plasma fractions and so on. We are using the LAL test to detect the presence of endogenous pyrogens in case of injectables. Also, you will be very interesting to know that LAL test is used to assess the food spoilage, water and air quality. Now, let us see what are the advantages of this LAL test, why we are preferring LAL test as compared to that of your rabbit test. The first advantage is it is less variable. Secondly, it is an in vitro test. As because the test is in vitro, so we are not requiring any experimental animals. So, automatically no animals are required. Now, as because animals are not required, so biological variation problem is overcome. We need not have to take the approval of CPC-SEA also. It is very easy to perform. Above all, LAL test is much more sensitive, less expensive and less time consuming. Due to these advantages, LAL test is more recommended in various pharmaceutical industries. So, but in spite of these advantages, this method has certain limitations. Limitations are the clotting enzyme is basically heat labile, it is sensitive to fluctuations of pH, and there is a possible interference problem specific for gram-negative bacteria only. Now, these are the list of biological products for which the pyrogen testing has already been replaced by the bacterial endotoxin test, commonly known as BET in IP 2018. The next slide shows the various blood products for which it is not replaced, but bacterial endotoxin test has been given as an alternative way. Instead of pyrogen testing, we can go for the bacterial endotoxin test or LAL test for these sort of pharmaceutical preparations. And with this, we are having these references. You can refer to those references for this particular topic of LAL test, and you will be seeing a very short video for pharmaceutical industries how the bacterial test has been carried out.
So first of all, to clarify, an endotoxin test is not a sterility test. I often get that question from our customers when they're performing a sterility test, and they'll ask, is an endotoxin test a sterility test? I have to explain to them no. But we do perform that test in conjunction with the sterility test because it measures the level of pyrogens within the formulation. That's particularly important when you have an intrathecal formulation that's going into the patient's spine. It can cause severe damage if that level of pyrogen is not measured appropriately. That's why we perform an endotoxin test. To explain more about the endotoxin test, I'll turn it over to our microbiologist here at Eagle.
Because of the significance of how harmful potentially bacterial endotoxins can be on humans and animals, especially if you're producing medical devices or injectables that come into contact with the bloodstream or spinal fluid, it's essential to test your sample for bacterial endotoxins. So they're a complex of lipopolysaccharides which are gotten when the cell lyses. Mostly they're gotten through contamination from water, so during prepping and making sterile injections as well. Sterilization is not the same as endotoxin free. Even though a sample might be sterile, it does not mean that it's endotoxin free. And because of the potential effects on human health and animals as well, you essentially want to make sure that your product is endotoxin free as well. Per USP, all category 2 injectables, that is compounded sterile preparations, and also those assigned with the BUD, or sterility testing as well, have to be tested for endotoxin. So during the test, we actually use an LAL test, which is a limulus amoebocyte lysate test. And essentially, the Limulus polyphemus horseshoe crab has these amoebocytes, which we extract from them. And upon contact with endotoxins, it causes a coagulation reaction. So the test itself, the instrument, measures when the coagulation reaction occurs. And with that, we can actually detect up to 0.001 EUs per mil, and making the test very specific and sensitive. We use the average body weight, the average patient body weight, and max dosage per hour to calculate the endotoxin limit allowed for a sample. And this is how we determine if it passes or fails as well.
For more information about USP85 endotoxin tests, feel free to reach out to us, 800-745-8916 or www.eagleanalytical.com.
And with this, we are coming to the end of our lecture today on LAL test. Thank you very much for your patient hearing. Hope you have enjoyed your lecture today. Thank you.